Mm. Good evening, everyone. Myself, Divya Chindan, and I'll be your host for today. So, before I start, I am thoroughly pleased to witness you all here in this workshop. It's my privilege to address you all on the behalf of our organization, Dollar Education. I welcome all the participants in the workshop. We are truly delighted with your presence. We all are gathered here today to attend this workshop, which is based on bioinformatics skills for vaccine designing. We as a company aim to achieve great heights. This workshop intends to resolve the practical hurdles faced during the vaccine development. So let's wait for 1 minute for others to join. Then we will start the session. Hello. Am I audible?
Yes, you are.
Okay, sir, I'll start the session again. Good evening, everyone. Myself, Divya, and I'll be the host for today. So, before I start, I am thoroughly pleased to witness you all here in this workshop. It's my privilege to address you all on the behalf of our organization, Dollar Education. I welcome all the participants in the workshop. We are truly delighted with your presence. We all are gathered here to attend this workshop, which is based on improving the bioinformatics skills by vaccine development. We as a company aim to achieve greater heights. This workshop intends to resolve the practical hurdles faced during the vaccine development. We'll wait for another 1 minute for others to join, then we'll start the We are now starting our session. So today's session is on preface of workshop and biology of vaccine design and case studies. This is the outline of the presentation. First, we'll be giving the introduction. Then the sir will be taking up few topics like biology behind the vaccines, COVID-19 successful vaccine design studies, the vaccine candidates, followed by the discussion and questionnaire round. So, our organization, Dollar Education, is an initiative to offer practical training on emerging concepts by making them affordable for all and then developing a consistent environment of growing together in the field. You can find us on social media with hashtag Dollar Education to keep us all in touch and also share ideas amongst each other. And we have named our organization because of its multi-faced meaning. That is, it is the currency name in around 20 countries. It is it is popular as an affordable symbol, and also it has been used as a variable in computer languages. The theme of the workshop is on designing vaccines. We have seen a lot in past 2 years, and it ultimately demands knowledge of bioinformatics from some perspective. For a very long time, different diseases have been eradicated, like smallpox and rinderpest, and a lot of these diseases have been eliminated by using vaccine shots. So, in the same way, you'll be learning various aspects of designing a vaccine. This is our work plan for the session. We have some time in between the lectures to give you all some time for practice and learn various concepts. Every session is for 1 hour, followed by a discussion. Any updates about the workshop will be posted on the website. For this workshop, we have Dr. Sandeep Dhanda, who is a bioinformatics research scientist at St. Jude Children's Research Hospital, Memphis, USA. Next is Dr. Swapnil Mahajan, who is a co-founder and a director of Deepnomics in Bangalore, India. And the last speaker is Dr. Rekha Kandia, who is an assistant professor at Barkatullah University in Bhopal, Madhya Pradesh, India. According to our records, we have registration from more than 7 countries, out of which more than half of the participants have intermediate or the beginners level of bioinformatics knowledge. Also, most of them are postgraduate, that is the budding level of their career. So we expect you all to make the best out of the workshop. You can post your queries in the chat box, and we request you people to be a bit interactive by replying to the questions which has been asked during the session. Also, we aim to provide the source of every information with proper references. There will be meaningful networking for your next career jump to appreciate diversity and multiculturalism. We make sure that all of you will understand the fundamentals of the topics to apply these skills into your projects. So is my screen visible?
Yes, it is visible.
Okay. Our first speaker for the day is Dr. Sandeep Dhanda. Today, I'm pleased to welcome Dr. Sandeep Dhanda to the inauguration of the Luminar program. He has completed his early education from government schools and colleges in Hisar district. Then he earned his master's degree in science from MD University in Rohtak City. He then moved to Chandigarh and earned his PhD degree in bioinformatics from a joint program of Institute of Microbial Technology, Chandigarh, and Jawaharlal Nehru University, New Delhi. Next, he pursued interest and received postdoctoral training from La Jolla Institute for Immunology, San Diego, USA. Currently, he is working as a research scientist at St. Jude Children's Research Hospital in Memphis City in USA. He's also a well-known peer- peer-reviewed and training young generation for the same with Publons Academy. He has an outstanding track record of scientific publication and also serves as academic editor for several reputed journals. Dr. Danda has published more than 50 articles in high impact journals and has developed dozens of heavily cited tools for vaccine design. Today, he is going to share his experience of vaccine design, and let's see what is known for this coronavirus vaccine. So far, he will be sharing his experience in epitope resource and prediction of T cell epitopes. So you're not audible.
Yeah, thank you, Divya. I think I can now share my screen and get started with my presentation. Is my screen visible?
Yes, sir.
All right. Thank you, Divya. Thank you for the great introduction. And this is about the preface of workshop and biology vaccine design and case studies. So in this session, I'll be talking about why we are planning this kind of workshop and what we are doing to understand the biology of vaccine design. And at the end, we'll be having some case studies so that we can discuss how these ideas were actually executed and how you can implement those ideas in your own research. So this is going to be the outline of my presentation. I will, as I said, there will be like 3 or 4 different components. The first component is of biology behind the vaccine. So what is a biology is underplaying that we are designing this vaccine and how we are coming up with a design. So with that, we'll start with what actually are the vaccines, how many different types of vaccines are there, and we are going to understand what do we call as immune response. And while we are talking about immune response, we must understand that immune response is a kind of um, a different entity for different type of vaccine design. That's why we'll be talking about the subunit vaccine and also what are the epitopes. Then again, we'll move on about the codon optimization and the determination of the structure. I will be just briefing about why we are putting all this concept in this workshop. And the next, we'll discuss how these ideas were implemented in successful vaccine design studies like that we are seeing from Pfizer vaccine, Moderna vaccine, and Covishield. I'm just picking three examples, but you will see any other vaccine. There is some component which was done from the design part, and it was done through the bioinformatics. So our stress is to show how bioinformatics is actually helping the process of vaccine design. And then next we'll touch upon this is the everything is done, then we're going to take these ideas and how we can take these ideas into our own projects, how we can initially create like our own project, and how we can publish it into journal with in silico ideas. And then again we are going to take it forward. If you have a wet lab, a collaboration, or have your own wet lab, how you can validate those findings. So we are going to talk about those essays as well, and at the end we'll be having some discussion question, and feel free to ask question along the way if you feel like that. So let's start with the biology part. Vaccines are doing great, and there's two diseases which are eliminated from the face of earth, like the smallpox and rinderpest. This has to be like deadly at the time they were diagnosed, or they were first diagnosed, but now it has been totally eradicated. And there are many other, like dozens of other diseases that are currently being eliminated. This include like polio, tetanus, flu, hepatitis, measles, pertussis, pneumonia, rotavirus, mumps, chickenpox, diphtheria. Why? Thanks to vaccines. It is because vaccines are really actually working. So our idea is to understand the biology, what are the component of these vaccines, how these are working, and how scientists are designing the very first component, the design part of the vaccine. And our stress is more about the bioinformatics. That's why we are going to stress about how bioinformatics can help you in the process of vaccine design. So I stole this poster from Dollar Education. So let's start with the very basic concept. We understand that we have a diverse group. We have very beginner participant. We have very advanced level of participant, and they have different bioinformatics exposure, and they are in under the different tiers of their academic career. They are graduate, undergraduate, or postgraduate, or postdoctoral. So we must start with the very foundational ideas. What is vaccine? So vaccine is made from very small amount of weak and dead germs that can cause disease. So any germ that is causing disease, some component of it could be a vaccine, or as a whole pathogen could be a vaccine. So it is actually if it is taking the whole component, we call it attenuated. In that case, it's a weak or dead germ. Or we can also take some component of it. In that case, we'll be calling it as subunit vaccine. So either taking the whole germ itself in dead form or in the weak form, or taking some component of the germ to elicit immune response, so that we can activate our body, so that we can prime our body in order to fight when we are actually encountering the real infection. So that means we are priming our body with small amount of germs or with dead germs, so that our body could recognize when the actual pathogen is attacking. So for coronavirus vaccine, we are taking some component of the coronavirus itself, then giving it to our body, so that our body start creating, like start developing our immune system in terms of antibodies, in terms of T cells. So how those things are going to play, that we are going to talk in further slides. And as it is mentioned by Divya, that we are providing trusted information. So everything that we are providing should be have a reference. So where we are picking this definition of vaccine, so you can find this vaccine definition from here. And this is a way of providing trusted information. So everything will be given a reference, due reference in this workshop. So talking about the components of vaccine, what actually constitute a vaccine? So when we have a vial or when we have an injection, what are the different components that have been added there? The first component is the active ingredient. That is the one that we were calling the germ part, the part of the germ which is like either the dead or the small amount of it, the bacteria or virus, and that is actually causing disease. That is an active ingredient, but it is not the whole soul of vaccine. It's also being supplemented by adjuvant. Adjuvants are like another entity to activate our innate immunity. And there are also some residual traces from manufacturing process. So while it is being scaled up, there are like some some traces of media or something will be there as well. And while we know that the vaccine is not going to be injected where it was manufactured, so it must have something to make it preservative or make it allowed to preserve or to stabilize, so that we can transport from where it is manufactured and from where it is being applied. And then it's also being added as some water to keep it soluble and to make it injectable. So these are the different components of vaccine. So the design part is mostly focused on active ingredient, though there is some research going on like how we can improve the adjuvants, but we will not be going in detail about the adjuvant in this workshop. Our whole theme is about talking about the active ingredient of a vaccine. And we'll not be talking about how you can have a better preservative, how you can have better stabilizer. So we will be talking about active ingredient. This is an idea to put ourselves into the right niche where we are talking about in terms of vaccines. And when we are having a good design, the next thing is to test it, to test whether it's really working. And when we say it's working or not working, we are actually testing for three qualities. We are checking for the safety. Safety means it should not create any other problem. It should be safe for anyone who might be having disease or not having a disease. So safety is the topmost priority for any drug candidate or for any vaccine candidate. The next is about the immunogenicity. The immunogenicity is something whether it is going to activate our immune system. So that's the by definition of a immune, like by definition of a vaccine, that it should activate our immune system. So if it is activating our immune system, that is immunogenicity. We are going to talk about what actually constitute an immunogenicity, what are different cell types that we consider as immunogenicity. And then the efficacy. The efficacy is something when we are injecting this into the individual, this might be very safe, like the water is very safe, but it is not immunogenic, or it is not having any efficacy against the germs, or some kind of like, let's say, toxins. The toxins are very immunogenic, they might not be safe, and they might not be very effective against to protect us. So these are the three basic fundamentals of vaccine design: safety, immunogenicity, and efficacy. And that's why there are like three different trials. The trial one is about the safety. I mean, obviously every trial considers immunogenicity as well. And the second trial is about immunogenicity. And the third trial, which is like a phase 3 trial, is mostly focused on the efficacy. So when we are talking about the efficacy data, let's say this company has a vaccine and they are saying like 95% efficacy and 90% efficacy, that is coming out of third clinical trial. So these are the different three trials that are actually constituting taken forward once we have active ingredient. So when we talk about active ingredient, so these are the different disease which has been like eradicated. This data has been taken from New England Journal of Medicine, published in 2013. It kind of shows that smallpox, which was killing around 4.8 million people in 20th century, killed 0 people in 2002. And thanks to vaccine, the same is true for polio. It was like killing around 1.6 million people, and now nobody is being actually the victim of this polio. The same way, we have a great progress in diphtheria, Haemophilus influenza, measles, mumps, pertussis, rubella, tetanus, which has been like deadly killer at the point they were initially diagnosed. But over the time we got vaccine, it might take decades to get a vaccine for them, but now we have a very effective vaccine, and now you can see that in the numbers we are saving literally the millions of people every year because we had vaccines. And how do vaccines works? So this is a textbook example, but there is some correction to it. The correction is on the left hand side, it's a antibody concentration. So it's measuring the immune response only in terms of antibody concentration, though the immune response is way more than antibody concentration. It could be T cell as well, it could be innate immunity as well. So considering this antibody concentration as the immune response in total, not just the antibody concentration, it says there are like two curves. The first curve is primary immune response. The primary immune response, when we are giving the initial exposure. The initial exposure means when we are giving that dead germ or the subunit, a protein part, and injecting into our body creates like start developing our immune system against it. So we have some sort of immune response. And that's why some people start feeling dizzy, some headache, or those kind of reactions, because our body start reacting to it. and we have some sort of immune response. But this immune response is not wiped away. It has some memory into it. So when we are actually having the real infection, let's say we have a vaccine for COVID, when we are seeing the real infection coming in, the real virus is trying to attack us, our body already has some antibody against it. Our body already has some sort of T cells against it. So when we already have some stored cells to fight that infection, those cells expand quickly. That's why the secondary immune response, when we are having the real attack, it is quicker. Now you see the time is less in case of secondary exposure, and we quickly prime our body and have a strong immune response so that our body prepare itself before the infection spreads. Is that clear? So it works in terms of primary immune response and the secondary immune response. In the primary immune response, we are priming ourselves, and the secondary immune response is something when we need it. The secondary inflammation is larger and quicker. That's why it's going to protect us from infection. Moving on about that, as I said, the antibody concentration, it was saying only the immune response in terms of antibody concentration, but that I also pointed this out, that immune response is way more than antibody concentration. It could be innate immunity, it could be adaptive immunity. So the antibody concentration is just this component on the right hand side, the B cell component. But we also have equally effective T cells like CD4 cells, CD8 cells. So these T cells and B cell joined together called lymphocytes. And these lymphocytes constitute the most important component of adaptive immunity. In parallel, we also have innate immunity. Innate immunity is something which is very general, like dendritic cell, macrophages, mast cell. These are not very specific in their action. But adaptive immunity is very specific against action, because the antibody or the T cell that are being kind of generated out of it are very specific to the sequences that they are generated against. So our immune response constitutes a couple of immune cells, and this could be further divided into innate immunity and adaptive immunity. Innate immunity is very generic in sense, and it's also rapid because it's very generic, like the skin cell, the mast cell, the macrophage, all these are innate immunity. And adaptive immunity is something very specific, and that constitute, like, these are the antibodies and these are T cells that are going to fight the sequences coming out of a virus or a bacteria specific to that particular pathogen. Moving on about the T cell and B cell, when we are talking about here, the T cell and B cell are the most important constituent of adaptive immunity and the immune response. The T cell and B cell has a fundamental difference. The difference is the antigen which is part of pathogen is directly recognized by B cells, and mostly those are present on the surface and being captured by the antibodies. So this is a mechanism of action for B cell. And same way we have T cells. The T cells do not act directly on the antigen, but these antigen need to be first chopped down and then processed through our molecule, which is MHC, major histocompatibility complex. So these MHC molecules should present them on the surface of a cell. Only then it will be recognized by T cell. So this is the fundamental difference between the B cells and the T cell in their mechanism of action. B cell recognize this antigen. The antigen is a part of germ directly, but the T cells do not recognize them directly. They need to be processed and presented. I will go into detail in upcoming slides. So while we are talking about this vaccine, we must understand that vaccine, there is a lot many research going on, and vaccines are now different types. That one is like live attenuated, where we started, like the tuberculosis vaccine, which was developed back in 1920s. So this is BCG, which is a bacteria itself, but it is attenuated. The attenuated means it has been processed through many passaging, like cell culture, culturing it once, then culturing it against, culturing it so many times that it has attenuated itself. Attenuated means it is no more kind of a threat or warning. And the same is true for the oral polio vaccines, or the measles, or the rotavirus, or yellow fever. In this case, we are injecting the live attenuated forms. We also have like another types where we create like inactivated or the killed antigen. These are the killed antigen which we are using in pertussis, the whole cell or the inactivated poliovirus. And in the next phase, we are going to touch upon the subunit, which is called purified antigen. The purified antigen is something when we are taking only a part of protein or just a protein or a peptide instead of taking the whole germ. And these has been really successful in acellular pertussis. Like pertussis, there are two different vaccines. The one is whole cell, the another is acellular, and these are also being kind of having a great record of success in hepatitis and pneumococcal disease. And we also had the fourth component, the toxoid. In this case, the toxin protein from the pathogen itself are being used as vaccine candidates. And the example is diphtheria and tetanus. All right. So as I said that there are different types of the immune cell. These are the B cell and these are T cells, and these are the innate and adaptive immunity. This is kind of a classical example. So bioinformatics is actually helping in these components. How? By summarizing what has been known to be recognized by B cell and T cell. What these cells are recognized in a germ, what these cells are actually looking for in a pathogen. So we know this because it has been already studied, it has already been reported in literature. So bioinformatics help in organizing that data. And bioinformatics has also some machine learning approaches where we can extend this known information. Let's say this is the pattern that our cells are recognizing. So can we extend this pattern to something unknown? So these are the component where bioinformatics is actually helping. The first is organizing what is known so far, kind of creating a complete picture of known knowledge, and then extending this known information to something unknown through the application of machine learning. And this is, I'm taking an example of subunit vaccine, because in subunit vaccine, and these are containing the minimal microbial element. That means they are only taking the smallest possible microbial element to activate our immune response. So we must understand what is that minimal microbial element that our body recognizes. And that is the exact definition of epitope. The epitopes are the smallest entity that our body recognizes. So the epitopes are mostly peptidic in nature, but there are also some non-peptidic sequences, non-peptidic epitopes. And these are identified through various assays. So there are some like B cell assays and T cell assays to identify those sequences in a pathogen. And if we try to look for what is the total space, how many epitopes are known, what our body is recognizing. So as I said, these epitopes are identified through experimental protocols. And then we have a bunch of people who report those in publications, and another bunch of people who read those publications and try to structure this data. See, this was the sequence tested and this was the immune response found. So try to connect a link. If there is a link between the sequence that they are studying and type of immune response that they are getting. So this is where this bioinformatics comes in the picture, that to connect those links, these were the sequences that were initially started with, and these were the immune response we kind of generated out of that. And that's why bioinformatics experts create the visualization platform so that you can see a bigger picture. And one such example is immune epitope database. It has millions of epitopes, and we are going to talk about the details of how you can find those sequences that are critical for critical to be an epitope, that are critical, that has already been shown to be an epitope. So those things are already been reported in publications and being listed and structured in IDB, that we are going to talk about the details of this IDB in our sessions, in day 2 session. And as I said, the bioinformatics works at two levels. The first, try to find out what is known so far and giving a comprehensive picture of this is what we know so far. And then take this information and connect to the unknown, to some sequences that is like a novel coronavirus. The sequences were totally novel. So how we can find out what are the sequences could be an epitope in novel coronavirus. So those kind of things could be done through machine learning methods. And these, as I said, T cells are fundamentally different from the B cell. The T cell recognize peptide when it is presented through MHC molecule, and there are some contact residue of the epitope or the peptide that interacts with both the T cell and the MHC molecule. That's why while we are talking about T cell epitope, we must understand that it is an epitope that had been under gone through processing. Then it will bind to MHC molecule. Then it will be T cell epitope, and after being a T cell epitope, there could be different arms of the T cell, the different differentiation part of the T cell. What type of cytokine are they inducing? Like Th1 immunity, Th2 immunity, those kind of thing will, these are the component at the T cell level. That's why when we're talking about the B cell, we only talk about whether the sequence is linear or whether the sequence is discontinuous. I need to plug in the charger. Yeah. So when we're talking about the B cell, we only understand that there are two different components, whether the sequence will be in a linear fashion or whether the sequence will be discontinuous. It will be one part located at, let's say, position 10th, the another is located at position 20th. So it's discontinuous in nature. So how we can design those kind of epitope and how we can consider those as a vaccine candidate. So this will be considered on day 3 and day 4 about the B cell. When we move about on the T cell, as I said, T cells are fundamentally different from the B cell. They must be presented through MHC. So we must understand how many different types of MHC molecules are there. So typically there are two different types of MHC, class 1 and class 2, and they have totally different ways of uh, presenting and creating epitopes. And that's why we'll be considering different days dedicated to understand about the MSc class 1 and to understand about the MSc class 2. And then we also, it's not necessary that as I said, it is a long process. Start with MHC processing, binding, and T cell epitope. So MHC binding, it just will consider the first two component. To understand the immunogenicity, the T cell part, we also need to understand the T cell epitope and the cytokine induction. Those things will be discussed on day 7 session. So this is kind of a theme and preface of the or design of this workshop, that we are touching from the very foundational things like the biology of the vaccine design and how these things are actually working in action, and what are the different components that are critical for a vaccine design. And these criticals are immune response, safety, immunogenicity, right? And how you can work around the immunogenicity, especially is the goal of this workshop. And the another thing that is coming out because of this RNA vaccine is the codon optimization. Now you know that there are only 4 different bases, and these 4 different bases are actually um, having a triplet that like arranged in a order so that they can kind of translate into an amino acid. But this there are some redundancy, like these 3 order or in this order or in this order will be creating serine, but the codon that constitutes the first is UCU or UCC is going to be different in different species. And this is an example from plant species where they have shown that certain kind of codons are preferred in one species than the other. And this is also becoming one of the critical component in vaccine design, especially with when we are talking about nucleic acid vaccine, let's say RNA vaccine and the DNA vaccines. This is really critical. This is because it helps to, when we codon optimize a sequence, we can also increase the expression. We can have the better expression when it is codon optimized. Let's say we are going to synthesize that in the cell lines. So it should be according codon optimized according to that cell line. If we are going to scale up using bioreactors in with some bacteria, so it should be codon optimized according to that bacteria. And the codon that we are using are different from are being used by viruses or bacteria, or maybe different cell line has different codons. So to to understand those what are the right codon that I should pick to come up with a design that help me to get better expression of my protein, or uh, to help me scale up my process. So those things will be considered in one of the session of codon optimization, and Dr. Rekha Khandia will be talking about how you can codon optimize your sequences. And the last thing that we are going to talk about in this workshop is mRNA structure. The mRNA structure, as I said, that these RNA vaccines are giving a new avenue, a new idea about the vaccine design. So this is a kind of publication on bioRxiv, and they said uh, they analyzed this 2p substitution in Moderna vaccine, and they said this Moderna vaccine is already been optimized for structure. So this RNA molecule, how they could be used as a vaccine, because they are so unstable. When we are injecting them in our body, they could be hydrolyzed quickly. So to make it stabilize, either they have to change certain kind of bases into some non-preferred bases so that they can stabilize it, or they have to change something. Uh, the structure should be somewhere that it is more stabilized than it is currently being found in native forms. So those things are being considered, how you can have a better structure that is more stable. The ultimate goal of RNA structure prediction here about the vaccine is providing a stability. So that when we inject into our body, it is not being chopped down or hydrolyzed quickly. It should have enough time to translate into protein, so that those protein are could be used to activate our immune system. The RNA is not going to activate enough immune response. That's why we need to have those kind of things in action. So this is all about why we have this theme, starting from let's say understanding the biology, then talking about what is known so far, how we can connect links, how we can predict B cell response, T cell response, and how we can find out codon optimized sequences, how we can have a better structure for RNA. So I have covered all different sessions and the ideology and the theme behind this workshop. Next, bioinformatics in vaccine design. This is what we have covered about the preface of the vaccine, preface of this workshop. Let's talk about how bioinformatics helped in current scenario, how computational immunology changed the face of COVID-19 vaccine development. So we got vaccine in a time of less than 1 year, and a big thanks to computational immunology, because that has provided the critical information that helped designing it quicker. Because let's say Moderna vaccine was designed only within a week, the Pfizer vaccine was designed only in 2 days, the Covishield vaccine was designed within a week. So how we could come up with such quick design, how we come up with such quick design, and this is really important to appreciate the computational immunology on these components. I will touch base upon 3 different vaccine as a case study, and the exact pinpoint to exact thing. This was the part done through bioinformatics. Starting with the Moderna vaccine. The Moderna vaccine is also well known for like 2p mutation. The 2p mutation means there are 2 proline uh, residues in the sequence of the spike protein that has been mutated here, changed here, to create a more stable molecule. And this is a kind of a news where it's like the tiny tweak behind the COVID-19 vaccine. So there was only 2 proline residue that were changed, and you can see those all the proline are residue are highlighted here in red, and only 2 of them were changed. And these 2 proline residue is coming out of a knowledge that were there in the lab, and the team initially created like hundreds of variants, and you cannot create hundreds of variant in the wet lab. So they created those variants in the bioinformatics lab, and then try to see which is more stable. And so they perform the stability analysis and the immunogenicity analysis. And this is an appreciation from the authors themselves. We created a vaccine molecule that could not have been created without structural information. So there was structural information playing well in the design part of Moderna vaccine. And while talking about the structural protein, we know that the protein structure and how we are going to have a better structure here in terms of proteins is a long topic, and we cannot cover the protein structure part here in this workshop. That's why we need to have another workshop on just the structural part of protein, where we are going to study how we can model structure, how we can dock certain compounds to it, how we can simulate them into certain environment. So those things is going to be a totally separate topic. But the idea here is to appreciate the structural biology information that contributed to the design of this Moderna vaccine. And this is another example from the Pfizer vaccine. Now you see, this is the real example, real sequence from the Pfizer vaccine. They have taken the virus sequence and the vaccine sequence. Now you see at certain positions, there are change in the nucleotide. You see CUU in the virus sequence itself is changed to CUG, though both of them kind of code for the same leucine residue. So they're creating the same leucine in the protein sequence, but the sequence at the RNA level was changed. That means they have done codon optimization, and codon optimization was done through many different packages that Dr. Rekha Khandia is going to talk about in this session, in her session. And at certain position, they also change this totally nucleotide, totally protein into a new one. Let's say it was coded for K in the virus, but it is coding for proline, like two proline are being changed here. So how they are coming up with this idea? So initially they said RNA molecular codon optimized, and there are some certain situation where they also changed the residue in the virus and that in the vaccine. And the reason behind it is either to have more stable or more immunogenic molecule, that we are going to talk about how you can create more immunogenic molecule out of a sequence. And the third part is Covishield vaccine design. In this Covishield vaccine design, this is a publication in Nature 2020, and where they have shown this was their design idea, the COVID-19 Oxford vaccine trial. They started with the SARS-CoV-2 protein, and then they kind of chopped out certain sequences, and they started with like the spike protein of the SARS-CoV-2 was taken from gene bank accession. The gene bank is again a biotech resource, and then they had kind of codon optimize it. So again, they are using the codon optimization methods to have their better design. And they also say we have chopped down certain residue from the start of this 5' end of this sequence. And this is because they can create a molecule which is again not being chopped down because of those 5' ends, and they cut it down. So all this design part that we are touching base upon, either it is Moderna vaccine, Pfizer vaccine, Covishield vaccine, these are the things where we have this information. But if you consider any other vaccine, you will see some component of it was done through Bioinformatics. And we are going to touch base upon how they did it. Then this is already been done. These vaccines are under many of them are under trial, many of them are undergoing in the real life. We are already been getting short out of it. So how you can take these ideas into your own publication, onto or into your own project? The first part is you can make in silico prediction and then run wet lab experiment. And this is what is done by these author in a publication Frontier in Immunology. They have taken 6 peptides and they run it through many pipelines, and they predicted whether they are going to induce interferon gamma response. That's why they picked it IFN epitope, which is one of the software that I will cover in immunogenicity day 7. So they predicted with it, and they found all the 6 epitopes that were shown to be positive for interferon gamma by the software were actually positive in the wet lab. So this is a validation, and they done it through FACS analysis, the cell sorting analysis. They say like in unstimulated, we are not seeing any immune cells, but when we have peptide P1, P2 stimulation with P1 to P6, we are seeing some sort of cell which are being stimulated. And that's why these servers are actually working. So you can first try to find out some candidates from your own project and run them through different pipeline. and then again validate them either with your own lab or with your collaborator, and you might get a good publication and your project could lead to a very successful design. So this is one example that you can do in your own research. The next is you can also create your own pipeline. So this is another paper that I'm citing, and they published in scientific report. This is about the MERS-CoV. The MERS-CoV is kind of a cousin of the SARS-CoV, which is the earlier version of it. And when they taken this MERS-CoV, they do the sequence retrieval and phylogenetic analysis, and then they perform the antigenetic kind of evaluation. They've done it by CTL epitope, STL epitope, IFN epitope, multi-epitope, and B-cell epitopes. And ultimately, they also added this multi-epitope construct to for strength, structure prediction, or for physiochemical parameter for antigenicity, and then they perform the docking. So this is kind of all in silico method. And ultimately, they come up out of this sequence from this virus MERS-CoV, they suggested these are the vaccine candidates. So there are set of tools that you can perform on your own project. You can start with your own sequence, then run this tool, analyze those data critically, and then report them into a nice publication. So these interesting findings, when we are really reporting something that is critical, that is important, this is acceptable by most journal. And I can say with my experience of being editor in 7 to 8 journals and reviewer in another 30 journals, I can definitely say when you have this critical set of pipeline created for your own sequence, you can get a good publication out of it. And this is what you can follow. This is a one pipeline, but there is also another pipeline you can also create your own. This is another pipeline where they started with again sequences, perform different ways of analysis, like the epitope prediction was done for T cell, MHC, and the B cell, and on the top of that they added antigenicity, allergenicity, toxicity to each of these epitope. Then they performed docking and ultimately constructed vaccines from different parts of the sequences. And this was all in silico. Then they also performed some structural analysis through docking or simulation, but some of them are doing this docking, but others are not doing it. That's why I'm stressing you can also create your own pipeline, and from this pipeline you can execute that it for your own project. You have a protein of interest, then you can run through different software, and from that software spit out, you can analyze it nicely and report in a nice publication. This is what you can do with your own pipeline. And this is the take home message and kind of my last slide, and I think I have enough time so that I can take questions. So our take home messages are: active ingredient of the vaccine is the most critical in design, right? And it should be safe, immunogenic, effective, and stable. Stable just for the transport purposes and to keep it freezer or keep it in the freeze or something like that. So it should be safe, immunogenic, effective. So we are going to talk most about the immunogenic part of it, because the effectiveness is tested only when we inject into individuals, but the immunogenicity could be uh, predicted by computer methods. So immunogenicity is measured by the antibody concentration and the T cell assays, and information on existing information, like the information on these epitopes, which are helpful for B cell response and T cell response, are really critical. And this computation method could really help in shortlisting vaccine candidates. Let's say you have a bacteria which has 3,000 proteins. The typical bacteria has 3,000 proteins. Out of the 3,000 protein, there could be only 1 or 2 or maximum 3 protein which could be good enough vaccine candidate. That's why those are the subunit vaccine candidates. So you can keep those proteins in mind, and then you can perform this analysis. And ultimately, you might suggest something that is very novel and could be useful in future vaccine designs. And with that, I'm done with my presentation and happy to take questions now. Feel free to connect me either on, you can search me on Google or find me on LinkedIn or Twitter with these handles. And happy to take questions now. And as I should also mention that I did not have any demo in this session because I want to touch upon the biology behind this vaccine. If we just directly jump onto the demo sessions, we'll be missing why we are doing this. The goal of this presentation to bring everyone into the same platform on the same page, that why we are running B cell epitope method, why we are running T cell epitope method, to bring everyone on the same page and to make you aware that these are methods available and you can do it even without having any coding exposure. And now happy to take question and to discuss more about the design part. And I think why we use water as ingredient, we can use other things. Yes, I think water is just one example, but there are also some media components or being treated like even in the media, there would be some water to make it stable or to make it soluble. So I wanted to ask that the codon optimization is only done for RNA vaccine to make them more stable. Uh, no, I mean, you see the vaccine from the Oxford, the Covishield is a protein vaccine, but it are also done the codon optimization beforehand. It is because when we are doing scale up, even the protein vaccines are being scaled up at the level of their RNA injected into a vector, into a bacteria. So if their codon is optimized according to that bacteria, it is going to help increase their expression of the protein and to easy scale up. Can we use this method for DNA virus? Yes, definitely. And you can pick many examples that DNA vaccines are processing the same way as the RNA viruses. What are the tool and the server to predict cytokine production? So for cytokine, so when we go after MHC class 2 binding, the T cell could differentiate into many arms. They could go into TH1. In that case, interferon gamma is more critical, and the server is IF and epitope. If they go into TH2 immunity, the IL4 is very critical there. So you can go IL4 pred. If they are going to the third arm, like let's say IL10 pred or more of a regulatory cytokine, so IL10 pred could be the one, IL17 pred is there, IL6 pred is there. So there are many different cytokine, but it will only be helpful when you know what kind of cytokine induction will be helpful in your vaccine design. Generally speaking, when we have intracellular pathogen, it is interferon gamma, and when we have extracellular pathogen, it's interleukin 4. Design a vaccine using bioinformatics. Yes, the percentage of accuracy, that's a really good question. I would say these are really accurate when there are different arms, let's say B cell and T cell. The T cell part are more than 85% accurate in general, but when we are like T cell epitope with MSC class 1 binding, they are 99% accurate. B cell part, we are not that accurate, but still we are heading in that direction. What is used as preservative? I'm not very much aware about what is being used as preservative. So you need to check your vial, and it will depend upon, I guess, it will depend upon what kind of ingredient is there. Let's say if there is RNA, it could be different preservative than when it has protein or when it has, let's say, the attenuated bacteria. What is general cutoff for B cell and T cell epitope? We are going to discuss the details about the cutoff and the threshold or anything of the B cell epitope in upcoming session. But in general, when we are using this software online, the default threshold will work for most cases until unless you have a greater reason to change that threshold. After modification, we have the same protein sequence. After modification, no, I mean, there are two different things here. When we are modifying the codon only, but it code for the same residue, we'll be having the same protein sequence. But when we also modify protein sequences, yes. Why we modify the RNA sequence? We can use the same. We can use the same protein, but let's say if you are this sequence is replicated in the virus, but we are going to scale up that in a cell line. So when we are going to culture that in a cell line or in a bacteria, our expression will be very less. To keep, and when we need a vaccine for millions or for billions of people, and the expression part is slow, we are going to lag behind in manufacturing process. That's why these are codon optimized. Sir, how do we go about shortlisting vaccine candidate for the in vivo testing per se? Yes. So how we are shortlisting? We are shortlisting based upon their immune response first, because immunogenicity is very important component here. So we are shortlisting based upon their immune response. Then we also shortlist them based upon how well they are optimized for scale up, how well they would do in stabilizing. So these are the things that you can add in advance for shortlisting before moving to in vivo testing. Best resource for whole proteome download of the organism. The best resource for the whole proteome is Uniprot. Uniprot is providing the reference proteome for all the organisms, and they are continuously updating it. As you give example, in that case, Covishield vaccine, they changed the amino acid using codon optimization. I'm not sure about they have changed the amino acid in the Covishield, but definitely they have changed it uh, in the Pfizer vaccine. And in COVID, so the tools that we are going to use are freely, they are that the tools we are going to use are freely available for academic purposes. But if you are using them for commercial license, there will be some nominal licensing fees in the IDB. And many of the tools that I can suggest you, those are free even for industrial purposes or commercial purposes. Should one go for in vitro testing of immunogenicity before selecting them in vivo? I think that that part is more of a wet lab design, and I don't go in detail because I'm not very confident about it. What should be the right way to move, whether it's in vivo first or in vitro first? But with my limited experience there, I would say generally like taking the blood sample out and doing the in vitro analysis can give you a good hint whether you should move in vivo or not. Or you can also unmute yourself and that we can directly chat rather than me reading from the chat. Anyone want to speak up? So what are the factors does the doses of the vaccine depend? Vaccine doses, the factor that depends again is, I would say when you say like, I would see this question as like whether you should need a booster dose or not. Those things are dependent upon how long our immune system works, like how good enough memory cells were created by the vaccine, how long the memory will be there. There are certain vaccine which we got only once and we have the memory cell for 20 years, for 30 years, or even for lifelong. Let's say for measles vaccine, you will see you got it when you were infant, and still you have those cells in your body. But there are other vaccine, let's say for coronavirus vaccine, the vaccine immunity is going down by the time. So it depends upon how effective memory cells were generated by the vaccine. By gene sequence analysis, can we predict whether it is? Yes, by sequence only we can make this prediction whether it is immunogenic or not, whether it is antigenic or not. And that's what we are going to discuss from tomorrow onwards in all the sessions. Can same epitope have be? Yes, same epitope could have sequences that are activating B cell arm and the T cell arm. In some cases, the epitope should be like 20 residue is good enough for a T cell response, but this 20 residue, there are some part which could activate B cell immunity. My question is how many rappel dose we need for COVID vaccine to get the best immunization against virus. So this how many doses would is a question, is still a question of investigation. And it is a question of investigation because there are different vaccine. Let's say for AstraZeneca, they only done it for one shot. So one shot was good enough in their trial, but then they said like if we get booster dose, you can even get better immunity. And for Pfizer and Moderna, they were tested for initially 2 doses. Now we were given third doses, and we might need even the fourth, fifth doses, but we don't know. It will depend, and it will be evaluated based upon the data that we see, how long it is going to protect us. That was side. How good enough memory cells were generated by the vaccines are critical to decide whether we will need booster dose or not. How the memory varies. The memory varies because the what could be the possible reason? And the tetanus vaccine memory is believed to be only 6 months, but other may last longer. The memory varies because the sequences or the type of the cells that activated by that vaccine, there are like 1,000 cells are activated by the vaccine. Out of the 1,000 cells, some of will go to the memory population. So how many cells are going for the memory population, and what sort of markers they are having when they transition to the memory cells? This is still a field under investigation. Nobody has a clear idea that these are the sequences that create memory cells and going to last longer. But research is going on in this section to understand how we can create memory cells. And it's true for MSC class 1 and class 2 to be activated by the same epitope. Yes, there are some sequences we have seen could activate both class 1 and class 2. Are antigenicity testing the same or different software? I mean, antigenicity is a generic term. The immunogenicity is the more specific one, I would say. The antigenicity, like whether it could, there are different software for antigenicity and immunogenicity. But immunogenicity is way more specific when we are talking about the T cell or the B cell. The antigenicity is a generic term for considering both the arms. How many sequences are initially identified for screening in silico from DNA sequence? How are the sequences initially identified for screening in silico from DNA sequence? I'm not completely understanding what do we mean by the how initially identified. Initially identified means, let's say for novel coronaviruses, we got the genome sequenced, and from the genome sequencing, we are picking up the sequences that this is a spike protein that seems to be more immunogenic than the others. That's why we are picking that up. Maybe that does answer your question. If we design a vaccine against any pathogen, we created a mutation in the protein, then how will we stop it completely? Yes, that's a problem with the vaccines, and that's why the idea of having the broad spectrum vaccine or universal vaccine is coming into picture. We are trying to see, can we have a vaccine that is effective against all the variants? But so far we even don't know how many variants would be there, and that's the problem. We are not having that highly effective vaccine against flu, because the flu is going to change every year, and when it has a new sequence, our previous vaccine might not work very well. So the idea for broad spectrum vaccine or kind of a universal vaccine is being studied, and there is no vaccine so far for especially for the viruses that could say it's a broad spectrum vaccine. So we saw that the trials are done in very short time, the development of vaccine for COVID. What's the difference between regular trial versus speed trials? The trials were done in quick, yes, in short time, that's true. But there were no compromise on the safety or immunogenicity or the efficacy of any trial. The trial were run in parallel. Let's initially we used to finish phase 1, then follow phase 2, then follow phase 3. But in the coronavirus cases, we were running in emergency. That's why we parallelly started the uh, phase 1 trial overlapping with the phase 2, overlapping with the phase 3, so that we don't wait for the good result from the previous trial. If we are having a bad result for the previous trial, all the 3 phases will be stopped, and those has been halted for many vaccines. That's why having those new design ideas in clinical trials helped this vaccine to come in a year. How about the doses of Covishield? But what is its what is of its concentration? I'm not sure about the concentration, but doses I see like there are two doses given for Covishield, and it will be evaluated over the time how these two doses are protective for, let's say, 1 year or 1.5 year, and then decide whether we'll need booster or not. Looks like we have a good discussion, but I want someone to speak out as well, not just texting. Would someone like to speak and ask their question? Uh,
Hello, sir.
yeah.
Sir, I just want your comments on the uh, discussion that Omicron is a milder variant. What are your views on that?
Um,
So the
see,
Omicron variant.
I understand, but I'm skeptical about commenting anything nonsense. But I have not read enough about the Omicron virus. Let me be frank about it. But what I have seen that it spreads quite easily than the other variants, and you see that millions of cases are coming every day, which has not been more than like 500,000 in any time in the past. That means it spreads easily. The other thing is infective, like kind of lethality. The lethality is low either because the vaccines are effective, or maybe the virus is mild, like is trying to adapt in our immune system, like the flu is not that lethal. And that could be the another thing to put into perspective. And the third thing is kind of our being immune from the natural infection, or being more protective about it, taking more care, or isolating the patient, doing quarantine properly. Those things could also be helpful. So I'm not really very sure about how to put into right perspective about the lethality in question.
Okay, sir.
Hello, sir.
Yeah.
So, what's the reason behind that some strains spreads very fast and some strains spreads very slowly, as we seen in COVID? And what's the reason behind the various scientists saying that Omicron, the third wave, is good for India? It will eradicate or it will end the COVID situation in world or India.
um, I mean, uh, firstly, like how they are having different variants and how there are different activity for different viruses. Different viruses has different nature because they have changed their genome, and when they change their genome, their kind of protein that they are synthesizing would be different, and that's why they might be having a different binding property with the human cells. Some of them might be having a better affinity with the human cells and going to infect even more, even easier. Or the other reason could be those sequences are more adapted to the human sequences. That's why they have better interaction and infect easily. And the second part of the question is like whether it will be good for India or not. I will not comment on that because I'm not aware how what are the all different consequences of this virus. So we cannot just assume without having a proper data in the same in the publications or being analyzed thoroughly that it will be good to eradicate COVID. Those are the things that are considered by, let's say, Sweden for in the first wave, that we are going for herd immunity. But later on, they realize that herd immunity might not be very good. They're kind of having natural immunity. So I'm not precisely sure how to answer that question in the right sense. As long as I don't have the enough data to speak on that.
Sir,
Yeah.
sir, why we should not give COVID vaccine to the people infected with COVID at that time? Will it worsen the disease or it will not create any primary response immediately? For that reason, we are not giving vaccine.
See, I'm not aware whether it is not allowed to give vaccine for the people who are already infected or infected at that point of time.
Yes. If I'm having a COVID right now, why shouldn't I be vaccinated?
I think it's
at
also
the same time?
because when you are already having the COVID, so your immune system is working in the same way that the virus will, the vaccine will work. So you are not going to have added advantage of the vaccine, but you might have something, let's say fever or headache or like those kind of mild symptoms out of vaccine being added to already uh, the symptom that you are having from the infection. So
Okay.
symptom wise, surely it will worsen, but immunity wise, it's not going to add up more. That could be justification of not giving when you are already infected at that point.
Okay. Thank you, sir.
Hello, sir. Sir, as we see that different variants are coming day by day, so do you recommend that different kind of vaccine should be developed for different variant?
Yes, that's why there are like more than 10 vaccines are approved worldwide.
Okay, sir.
And we don't know, like, let's say for Delta virus, vaccine 1 was more effective, and for Omicron, vaccine 3 could be more effective, and might be there could be more variant where another vaccine would be more effective.
Okay, thank you, sir.
You're welcome. All right, so I think we had a good discussion, and we can continue this session tomorrow when we are going to talk about the resources about the epitopes and how we are going to find what has been discovered so far about the epitope and the sequences. It was really nice presenting here and have a great audience like you. Thank you all from the bottom of my heart. And thank you, Dollar Education, for giving opportunity to talk about my experience and exposure in vaccine design. See you guys tomorrow.
Thank you, sir. Thank you so much.
Thank you, sir.
Thank you, sir.